Hi everyone, and welcome to this presentation on the use of green fluorescent protein in biology. Today we will discuss the purpose of the green fluorescent protein as a technique and what it is used for. We will also discuss the history and development of the technique and how it works, as well as its current applications, advantages and shortcomings of the technique, and also what the future holds for applications of the green fluorescent protein.
Found in the Pacific Northwest of America, the Aquaria victoria jellyfish was the first observed source of the brightly glowing green fluorescent protein in 1962. Osamu Shimomura, a Japanese research assistant whose remarkable success in research into the bioluminescent light production of the Japanese sea firefly Cypripedinia, ignited his lifelong passion for the study of bioluminescence. This earned him an invitation to study Aquaria victoria at Princeton University in 1961. Later, Martin Chalfie, Roger Chen, and their associates continued on his discoveries of GFP. As Professor Gunnar von Hein stated in his announcement of the 2008 Nobel Prize for Chemistry to Shimomura, Chalfie, and Chen, the protein has become one of the most important tools used in contemporary bioscience and been imperative for bioscience to map the role of different proteins, with researchers developing ways to watch previously invisible processes within the body. Shimomura's research involved collecting the jellyfish from the abundant natural resource by the laboratory's dock at Friday Harbor in Washington each morning and evening. The near-transparent jellyfish are shaped like hemispherical umbrellas, measuring approximately 3 to 4 inches in diameter and weighing about 50 grams. The light granules of interest are evenly distributed along the edge of the umbrella and were cut from the jellyfish by hand using scissors. The rings were then squeezed through a round gauze to produce a dimly luminescent liquid called squeezate. However, when the squeezate was diluted with water, its luminescence significantly increased for a 5 to 10 minute time period as the granular light organs were cytolized. Isolating the luminescent substance from the squeezate proved difficult and was not able to be extracted in the same way previously done by Cypripedinia, by exhausting or eliminating the cofactor involved in light emission. Extraction of the substance was eventually achieved when Shimomura lowered the pH of the squeezate, filtered the solids out, producing a nearly dark filtrate which regained luminescence upon neutralization when sodium bicarbonate was added. His next step, adding a small amount of seawater, had remarkable results with explosively strong light being emitted. This experiment proved that a component of seawater was the activator, leading to the quick discovery that calcium was the activator and that EDTA is a better inhibitor than acidification during extraction. Shimomura now had a workable way of extracting the luminescent substance from Aquaria victoria. It was discovered that 2 proteins are used by the Aquaria victoria to make its green light. Green fluorescent protein was found to be a companion protein to a calcium-activated photoprotein that Shimomura named aquarin. Aquarin generates a high-energy blue light when in the presence of calcium, with this chemiluminescence peaking near 470 nanometers. The low-energy green bioluminescence has an emission peak of 508 nanometers, with an excitation peak near the emission peak of aquarin. Therefore, the blue emission of aquarin is converted by green fluorescent protein to the fluorescent green seen in intact cells. This radiationless process of absorbing and re-emitting light energy is fluorescence, hence the second protein's name, green fluorescent protein, or GFP. Due to the extremely low content of green fluorescent protein and it being extremely laborious to extract, GFP was relegated to a side project while samples were accumulated and further research was done on aquarin alone. Further experiments weren't done with GFP until the early 1980s, when Douglas Prasher sought to utilize GFP's fluorescence as a tag. He believed by inserting the GFP gene at the end of the protein just before the stop codon, then the protein of interest would be made with GFP tagged on its end with the green fluorescence showing whence it was made and where it moved within the cell or even the whole organism. Prussia, however, had little success with inserting the GFP gene he isolated and getting the protein to fluoresce. Prussia's GFP DNA was later given to Marty Chalfie in 1992. His colleague Gia Erskine successfully inserted the gene by using primers that copied only the coding sequence rather than including the additional base pairs before and after the gene as Prussia had done. A new technique called polymerase chain reaction, PCR, was used. This amplified the DNA, enabling the GFP to fold correctly and emit the desired green glow. This was done without the use of restriction enzymes or any additional jellyfish proteins, which was previously thought to be needed for GFP to fluoresce. This crucial breakthrough of cloning the gene and successful expression of the gene in other organisms proved that the GFP gene contained all the necessary information to carry out post-translational synthesis of the chromophore. When GFP cDNA is expressed in a nematode as a reporter gene using a promoter utilized in only a few nerve cells, the GFP fluorescence extended throughout the cytoplasm of the nerve cells, illuminating the axonal processes and cell bodies. Next in the development of green fluorescent protein, Roger Chen, along with his team, worked with expressing GFP in yeast and modified it to fluoresce blue, yellow, cyan, as well as a brighter GFP called enhanced GFP, or EGFP. Later, more colours were developed called the M-fruits. EGFP is now the most commonly used fluorescent protein. EGFP was developed to overcome the limitations of Aquaria GFP, including low folding efficiency when expressed at 37 degrees. As Aquaria Victoria found in cold water, the mature protein is most efficient at temperatures well below 37 degrees. Other limitations: low fluorescence when excited with blue light, and in several mammalian cells the gene had poor expression. By substituting the PHE64 with the LEU, folding was improved at 37 degrees, and a substitution of SER65 to THR caused a 35 times brighter luminescence than the wild type. Mutating GFP has produced proteins with altered excitation and emission spectra and increased fluorescence intensity. These altered proteins are more sensitive markers, have the ability to double and multi-label, and also brought an understanding of fluorescence molecular basis. The various mutations achieved demonstrated that GFP's fluorescence properties can be altered by mutating the GFP gene sequence within and outside the chromophore. Experiments have also revealed that reagents can be obtained to extend GFP's usefulness as a biomarker, particularly using blue emission GFP for double labeling. The chromophore of GFP is particularly unusual as it is formed from the primary amino acid sequence of the protein. Shimomura found that the chromophore is a 4p hydroxybenzylidene imidazoline 5,1 attached to the peptide backbone through the 1 and 2 positions of the ring. Spectral characteristics can be altered of GFP by changes in pH, protein concentration, ionic strength, and temperature. Fluorescent properties of GFP have been demonstrated through mutation experiments to be dependent on the protein sequence within and outside the chromophore peptide. GFP has been shown that it can be used as a non-invasive way to monitor gene expression in vivo through the mentioned experiments, and does not require cell permeabilization or toxic detection methods, and can be made in many cell types and cellular compartments. Experiments have shown that the excitation and emission spectra is the same as AGFP produced in E. coli and native GFP. GFP expression can be used to mark specific cells as it illuminates the growth cones at the end of the axons, allowing the details of axon targeting to be followed in live animals and monitor morphological changes in living cells. Proteins can be tagged by GFP, allowing the study of their subcellular location. GFP is superior over antibodies in specificity, and cross-reactivity does not occur. GFP can be expressed in a variety of cell types as a single protein or as part of a protein fusion, with the most successful experiments having used strong promoters. fluorescence becomes stronger with illumination.
Since the discovery of green fluorescent proteins, molecular biology and modern science has seen incredible advances from a range of applications. The applications that we will discuss today range from protein tagging, monitoring of gene expression, and biological screening. For the generation of green fluorescent proteins bioluminescence, no other substrate or cofactors are required. It is independently generated. Under laboratory conditions, exposing green fluorescent protein to an ultraviolet source is an effective method of mimicking the natural transfer of energy, which enables activation of the protein's bioluminescence. One of the most significant applications of green fluorescent protein is its ability to enable protein tagging in both in vitro and in vivo studies. Protein tagging is achieved using standard subcloning techniques where green fluorescent protein and a cloned gene of interest are fused together and inserted into a cell by way of transient or stable expression. This was first enabled when biologists heterogeneously inserted complementary DNA from green fluorescent proteins into living bacterial cells. Protein tagging was initially applied by scientists using conventional fluorescent microscopy at a cellular level to localize and track proteins within organisms. This visual marker is more commonly used in eukaryotic organisms than prokaryotic organisms, due to lack of constructs within the cell. Improved isoforms from mutational analysis of the protein's chromophore structure allowed researchers to experiment with emission of alternate colors and brighter spectrums. All major biological model systems now essentially have a compliant isoform of the green fluorescent protein. The first particle to be tracked using green fluorescent protein were ribonucleic protein particles in the fruit fly. From hereafter, further applications enabled the visualization of several intracellular compartments. These include visualization of cellular organelles and their properties, the mitotic apparatus, elements of the cytoskeleton, protein trafficking, and also secretion events. Protein tagging has also allowed scientists to observe transcription and pre-mRNA splicing. During interphase within the cell nucleus, scientists have been able to make these observations due to fusion of green fluorescent protein to the pre-mRNA splicing factor. Applications of the green fluorescent protein have also enabled visualization and tracking of DNA sequencing in mammals, bacteria, and even yeast. There are, however, risks when working with green fluorescent protein that are important to be aware of. As mentioned previously, there is a risk of green fluorescent protein causing free radicals within the implanted cell, which can lead to nucleus and DNA damage, and also long exposure to UV light can cause cell cycle arrest and, worst case, the death of a cell. To avoid this, care must be taken to ensure the living cell is not overexposed to the UV light. Another major application of the green fluorescent protein as a vital reporter molecule is its use in monitoring gene expression in single living cells and tissues. Monitoring of gene expression is enabled when the green fluorescent protein's N or C terminal fuses with a gene of interest. The subcellular location of these proteins is then tracked by distinct staining patterns observed using fluorescence microscopy. Fast transcriptional activation processes are quite difficult to achieve due to the slow nature of the green fluorescent protein folding into its active form. The last application that we will briefly discuss today is green fluorescent protein's role in biological screening of genes and even drugs in living organisms. For biological screening to occur, an expression vehicle must carry genes from the green fluorescent protein which have been co-expressed with a second gene of interest. The green fluorescent protein signal is then followed by visual screening. This type of screening can be used to monitor genes carried by stable cell lines and also allows for rapid cell enrichment. Green fluorescent proteins have also been used in living organisms to monitor the fate of particular cells. This can be used for applications such as tracking viruses to even embryos in mothers.
So on this slide, we're going to look at the advantages of GFP compared to other reporters used to track protein and gene expression in cells. The biggest advantage GFP presents is its ability to be used both in vitro and in vivo using microscopy. This allows biological events to be observed in a dynamic manner without altering the biological processes of the cells being examined. Another key advantage of GFP is that the fluorescence only requires a simple blue light to be detected rather than other potentially invasive treatments in which the cells are exposed to. No added cofactors or substrates are required for its detection also. The next point is that it is quite a small molecule with a monomeric structure that allows it to diffuse easily throughout cells and outline their structure. More advantages include the fact that GFP fluorescence is generally not affected by changes in pH and temperature or the presence of denaturants and enzymes, for example. Lastly, in addition to being utilized for qualitative analysis, GFP is also useful for quantitative analysis using flow cytometry. This allows accurate quantification on a cell-by-cell basis. So on this slide you can just see an example of GFP's biggest advantage, which is the use in live organisms, also known as in vivo. So you can see a range of species here, all expressing GFP safely, from all the way from, on the top left, a nematode, all the way through to a rabbit. And on the right you can just see the cellular level of that expression there as well. Despite revolutionizing biology and biomedicine, GFP has several minor disadvantages. One such disadvantage is the use of GFP under hypoxic conditions, which commonly occur in cancerous tumours, for example. Researchers found that with lowered oxygen conditions, the natural fluorescence of GFP decreased, and in anoxic conditions it disappeared almost completely, possibly due to GFP requiring molecular oxygen for formation of the fluorophore, meaning it is relatively unsuitable for use as a gene expression reporter in tumour conditions. And on the bottom left there's just a little diagram where you can see as the oxygen increases, the fluorescence increases, and as it decreases, the fluorescence also decreases. So another possible disadvantage of GFP is the inability for it to be secreted out of the cell, making it challenging to quantify gene expression. This limitation, however, has been successfully overcome by the enhancement of GFP to form eGFP, or enhanced green fluorescent protein, with the addition of a single peptide sequence, thus allowing it to be secreted out of cells, making this not really a limitation to consider since it has been overcome. But it's still worth mentioning. The next point that may be considered a limitation is the variability of GFP expression across different cell types. Researchers found that there is actually a high degree of variability in GFP expression across different cell types, where it may be expressed in higher quantities in blood cells, for example, than the cells of organs or other tissues. This occurred even when using several GFP transgenic mouse strains, which are thought to express the protein ubiquitously across every cell. And the last point, recently there has been some new research linking enhanced green fluorescent protein, or eGFP, expressing cells in mammalian and bacterial cells to the production of free radicals, also known as reactive oxygen species. And the ones that they have found have been produced are superoxide and hydrogen peroxide. You can see them there on the little picture on the right. So this can possibly cause tissue abnormalities and cytotoxicity in the cells, and could possibly complicate validation in experiments conducted using enhanced green fluorescent protein. This slide will address the future applications and future research concerning GFP. Firstly, there has been some new research into the use of modified GFP as a pH biosensor. Researchers have modified the protein by adding fluorotyrosine residues into the fluorophore, which then causes an alteration of the fluorescence and a change in the pKa of the phenolic protein of the fluorophore. With further development, researchers think this refinement may open the door to the potential for modified GFP as use as a pH sensor or biosensor. Now the second point is about the development of other fluorescent proteins. So we know from modifying GFP, the colours cyan, blue and yellow have been further refined and developed, and from other organisms such as corals, the orange, red and the close to infrared, far red fluorescent proteins have been created. These red and far red fluorescent proteins are ideal for use in mammalian cells due to minimizing light absorption by tissues and autofluorescence. The use of these different color fluorescent proteins is proving useful in cancer research. For example, a transgenic GFP mouse can be transplanted with cancer cells expressing red fluorescent protein, enabling the interaction between the host cells and the cancer cells to be visualized in vivo in real time. Researchers have also been able to modify cancer cells with two different fluorescent protein colors. For example, green fluorescent protein in the nucleus and red fluorescent protein in the cytoplasm to observe subcellular dynamics. Possible future applications in this area are techniques such as fluorescent-guided surgery, due to the ability of fluorescent proteins to be inserted into tumours using viral vectors in vivo. So here in these pictures you can see examples of these applications taking place. On the left you can see a GFP mouse with a red fluorescent protein illuminated tumour, and on the right you can see an interaction between the host immune cell in green engulfing the cancer cell in red. So you can see how using multiple colours of fluorescent proteins is quite useful for the visualization of malignancy. Lastly, a big area of future research for fluorescent proteins concerns the red and far red varieties. All naturally occurring red fluorescent proteins have a tetrameric shape, which isn't really ideal for applications in cell biology. This is creating a need for alteration of these red fluorescent proteins to make them monomeric instead, where they will be more suitable for biological applications and easy diffusion throughout the cells. This is proving challenging, however, with attempts at monomerization creating dimmer and more blue-toned red fluorescent proteins. In fact, only 4 have been successfully monomerized, meaning there are many more remaining which are awaiting monomerization. So this is really the future of fluorescent proteins, is these red, orange and red proteins.
Thank you for watching this presentation, and we hope you enjoyed as we delved into the world of the green fluorescent protein in cell biology.